All right, so we're looking at CFOS signaling, right? And we don't want to pay attention necessarily to this, which is something different. We want to induce the CFOS gene, right? We want to make CFOS mRNA to make the CFOS protein, which then works as a transcription factor. And so we said that there was a lot of pathways that converge on the CFOS gene, right? So the CFOS gene has multiple mechanisms that allow it to be regulated, right? Multiple binding sites that allow for activation of a different transcription factor. So how do we say that? Maybe multiple binding sites that mediate transcription. Transcription, not translating anything yet, but transcription. So one is targeted by CREB. So there's a regulatory region of the CFOS gene where CREB can bind. There is a regulatory region that is affected by downstream of RAS. There is a regulatory region that responds to calcium and cyclic AMP. So these can all help regulate induction of CFOS. And so let's take a look at the diagram, right? The diagram is slightly different, but the idea is the same, right? So growth factors are one thing that we know can regulate CFOS induction. And so, again, there's probably members of this pathway that are missing in here, but the end result is to activate CREB, right? Neuromodulators that activate G-protein coupled receptors, right? Elevate cyclic AMP, as we mentioned, and cyclic AMP can activate PKA. PKA can phosphorylate CREB and allow it to bind, so itself working as a transcription factor to allow CFOS to be transcribed. And then calcium, right, through voltage gated channels can have an effect on transcription factor binding and may have a binding site itself. The whole point, right, is this: multiple converging pathways that regulate how much CFOS protein will be present. Okay. Next. As we reach the end of this chapter, I think maybe the best way to summarize it is to look at 3 different examples of neuronal signal transduction, right? That's what this whole chapter has been about. So 3 examples. Neuronal, oh, I spelled that wrong too. It's getting late. Neuronal signal transduction. So we'll go through 3 different pathways. The first pathway that we'll talk about is through NGF and TrkA. NGF is a protein that's required for differentiation survival. So this thing is a protein for differentiation, survival, as well as sympathetic connectivity, so those fight or flight responses. In order for neurons, right, pre and post ganglionic sympathetic neurons to talk to each other, they require NGF. Neurite growth factor is what it stands for. sympathetic connectivity. All right. So how does this work? Well, NGF is the signal, right? This is the signal that works to induce change. How does it do that? Well, let's look. It binds to a receptor, right? A receptor on neurons, a receptor tyrosine kinase, specifically the TrkA receptor. So step 1 here, I already used 1, so let's do letters A. NGF binds its receptor. This receptor tyrosine kinase, specifically TrkA. What you see happen next, right, is this receptor comes together. It dimerizes, right? So these subunits of the TrkA receptor are typically separate, and they come together, right? So we call that dimerization, when two protein subunits get together. So the receptor dimerizes. Which basically means the two parts come together. Dimerization. Those two parts are coming together, right? And that there's this what's called then autophosphorylation, when each of the two parts phosphorylates each other. So we'll say that each portion of the receptor each part of the dimer phosphorylates itself. It's Ceph. Yeah, you get the idea. I'll leave it. This triggers a cascade via RAS. So what will happen is the small G protein RAS will be activated. Okay. And RAS is able to activate multiple protein kinases, as second messenger targets, multiple protein kinases now are turned on. These kinases can move into the nucleus and phosphorylate Krebs. All right, so they phosphorylate Krebs. And this leads to essentially development, that the neurites will grow outward, right? So early neurons are called neurites, and their axons will grow, and maybe they're not differentiated. Maybe we know that we have a neuron, but we don't know what type. Is it GABAergic or cholinergic? Right? So this leads to neurite outgrowth, right? Development of the axon, of the dendrites, all of those things, as well as differentiation. What type of neuron will it become? Okay, so that's one pathway that this can follow, right? Is through activation of RAS. So I'm going to leave some of this up here, but I want to talk about where this process sort of diverges. I'm going to leave up the portion where the receptor tyrosine kinase dimerizes and phosphorylates itself. Another direction this pathway can go is to activate PLC, right? So this receptor tyrosine kinase, because it's phosphorylated, can activate different enzymes, right? And one of those is phospholipase C, so PLC activated. We've been over this a few times, but this increases the amount of diacylglycerol in an inositol triphosphate. So increasing DAG, diacylglycerol, and increasing IP3, right? Because remember, phospholipase C cleaves PIP2 into these two products, okay? IP3 triggers calcium release from the ER, right? Because it binds to IP3 receptors and allows calcium to flow out. So this IP3 causes calcium release from the ER. Well, now that we've got calcium present, now that we've got diacylglycerol present, so DAG, calcium, that triggers the activation of PKC. What does this lead to? Neurite outgrowth, neurite differentiation. Or neuronal differentiation, I should say. So outgrowth, the same thing that we saw before, right? Outgrowth and differentiation. Differentiation is what that says, believe it or not. At the very bottom of the slide, it's tough to write. Okay? Now, yet there's another pathway that activation of TrkA receptors kicks off. So again, I'm going to leave up the dimerization portion. And we certainly didn't talk about every possible kinase, right? And so another pathway that this could activate are these that maybe kinases that we haven't talked about, right? So dimerization of the TrkA receptor and autophosphorylation lead to activation of other kinases. Right, and so we see here Pi3 kinase. These other kinases, like Pi3 kinase, inhibit cell death. Right? And so in that way, it promotes cell survival. All right, so that's just one of really multiple ways, right? So we've got the Pi3 kinase pathway, cell survival. We talked about what happens with RAS. We talked about how phospholipase C gets active. These two pathways converge to promote outgrowth and differentiation, whereas other kinases promote survival. Okay, the second specific signaling pathway I want to talk about is the pathway that involves something called LTD, or long-term depression, in what are known as Purkinje neurons. So we're going to have to talk a little bit about some anatomy first before we get into this. So this process, right, is happening within the excitatory synapses of these cerebellar Purkinje neurons. So where are we? Specifically, we're within the cerebellum. And there are these cells, there are these Purkinje cells that form enormous dendritic branches, right? Their arborizations are huge, okay? And so particularly, right, these Purkinje cells and the connections that they make that are excitatory connections with what are called parallel fibers that emanate from granule cells. So these are both types of neurons. Okay, so parallel fibers. So look here, right? Is this in black? Right? This is the Purkinje neuron. And this in blue is the parallel fiber. And so we're going to look at these synapses that are in yellow. And so I don't have yellow, but you can see these are the synapses that we're going to be talking about. Okay? That's our orientation. And if we look at this diagram here, right? So this is the parallel fiber, and this is the Purkinje neuron. Okay, so I'll come back in the next video and talk about what happens here as a mechanism of long-term depression.